The next thing that we want to go ahead and consider is something called diffusion and osmosis. These processes are going to go ahead and be a very important part of structures or substances moving inside of and outside of cells. For the body to function, you're going to have a lot of movement across the plasma membrane. And the processes of diffusion and osmosis are going to go ahead and accomplish that movement across membranes. Diffusion can be defined as the movement of molecules down their concentration gradient. When we're talking about diffusion, we're talking about the fact that substances are going to move from where they're in high concentration to where they're in low concentration. As an example, let's go ahead and consider a sugar cube being dumped into a beaker of water. After we go ahead and drop that sugar cube into the water, what's going to happen is that individual sugar molecules are going to begin to break off from the lump and move away. These sugar molecules are moving from an area of higher concentration in the lump to an area of lower concentration away from the lump. More and more sugar molecules will move away and randomly bounce around until ultimately, eventually, all of the sugar molecules will become evenly distributed throughout the water until we reach a point that is referred to as equilibrium. Now, osmosis is not exactly the same as diffusion. Diffusion is simply, again, the movement of substances from high concentration to low concentration. But osmosis specifically is dealing with the movement of water. It's the diffusion of water through a semi-permeable membrane. And what we're dealing with here actually is the fact that water itself is moving. And water is actually moving from where water is in high concentration to where water is in low concentration. When we're dealing with osmosis, we're dealing with the fact that we have something called solutes. Solutes are going to be substances that are dissolved in a solution. For instance, we might have water. Water being a solvent into which solvents can be dissolved. When you dissolve your solutes in a solvent, they form a solution. We can go ahead and have three types of solutions. Number one, we can go ahead and have a solution that's called hyperosmotic. Number two, hypoosmotic. And number three, isotonic. A hyperosmotic solution is going to go ahead and contain a higher concentration of solutes than the cell. In this particular case, you're going to have, for instance, a cell being bathed in a solution, and the solution in this particular case is going to be more salty or have higher solutes than the cell itself. The second scenario is where we have a hypoosmotic solution containing lower concentration of solutes than the cell. In this particular case, we have a cell being bathed by a hypoosmotic solution. The solution surrounding the cell is going to be less salty or have lower solutes than the cell itself. And finally, we could also have something called the isotonic solution, which contains equal concentrations of solutes as the cell. In this particular case, a cell is being bathed by a solution that is going to have the same concentration of solutes, both in the cell and in the surrounding solution. Let's take a look at an example of osmosis. Here what we have is a beaker of water, and we also have a membrane dividing these two halves of the beaker. It's called a semi-permeable membrane, meaning that some things can move through and some things cannot. Specifically, water molecules are relatively small, so they're capable of moving through, but larger molecules probably will not be able to pass. Now, diffusion causes water molecules to distribute themselves equally on both sides of a semipermeable membrane when you only have water molecules on both sides. In figure number 2, what we see is a situation where we have the same beaker, but in this particular case, we have the addition of urea molecules to one side of the semipermeable membrane. On one side of the membrane, we're going to have only water molecules, but on the other side of the membrane, we have both water molecules and urea molecules. In this particular case, addition of solute molecules that cannot cross the membrane, which would be the urea molecules, reduces the number of free water molecules on that side as they bind to the solute. If you go ahead and consider concentrations on the left-hand side and the right-hand side of that semipermeable membrane, you've got a situation where water is going to be in higher concentration on the left-hand side of the membrane than it is on the right-hand side of the membrane. And what you're going to have occur is a situation where osmosis, or movement of water, will occur where water molecules are going to move from where they're in higher concentration to where they're in lower concentration. The water molecules are small enough to cross the semipermeable membrane, whereas the urea molecules are not. Notice that on the left-hand side of the semipermeable membrane in the beaker, you can see that the solution is labeled as hypotonic. This is in contrast to the right-hand side of the beaker on the right-hand side of the semipermeable membrane where the solution is labeled hypertonic. The words hypotonic and hypertonic can also be used interchangeably with hyperosmotic and hypoosmotic. In this particular case, we have on the left-hand side the hypotonic solution because it has less solutes. It just has water. And in contrast, on the right-hand side of the beaker, we have the hypertonic solution because it has higher solutes, with urea being the solutes. At number 3, what you're going to go ahead and see is a situation where we're going to have movement of water molecules. These water molecules are going to go ahead and move from the left-hand side of the beaker to the right-hand side of the beaker, crossing the semipermeable membrane. The water molecules are going to go ahead and move from where we have a higher water concentration to a lower water concentration, based on the fact that a significant portion of that area is composed of urea molecules. In addition, what we're going to have is a situation where the water molecules are moving from an area that is going to go ahead and be lower in solute to an area that's higher in solute. So the water itself is going to go ahead and move essentially to wherever there is more stuff. Again, osmosis is simply a form of diffusion. Diffusion is where we're going to go ahead and have movement of molecules from higher to lower concentration. That diffusion can be of any type of molecule. Osmosis specifically pertains to the movement of water. Water itself is going to move from wherever water is higher to water is lower, or to wherever you have more solutes. Now, movement of water into a cell can create something called osmotic pressure. This can ultimately cause a cell to swell and burst. Here we have the same cell under different osmotic pressures. Number 1, the cell on the left is a typical red blood cell. This red blood cell has a typical donut shape. However, this particular red blood cell has been placed in a situation where it now has increased its water volume inside of the cell. It was placed in fresh water, and fresh water entered the cell from an area where it was in higher water concentration to where it was in lower water concentration. As water moves into the cell, it's going to swell and could again potentially burst due to higher osmotic pressure. Now, as I mentioned, things are going to go ahead and have to move into and out of cells. And we're going to go ahead and have things moving into and out of cells that are going to go ahead and be bulky or particulate, as well as things that are going to go ahead and be liquids. Bulky substances are contained within vesicles as they are moved into and out of cells. And the diagram below is going to go ahead and illustrate movement of substances from the inside of a cell to the outside of a cell. When you have substances moving from the inside of a cell to the outside of a cell, that's referred to as exocytosis. What we have here is a situation where things are exiting the cell. Exit, exocytosis, which is going to be discharge of substances from vesicles at the inner surface of the cell. The bottom half of this diagram represents the inside of the cell. The top half is going to represent outside of the cell. And you can see in white that we have the plasma membrane separating the inside from the outside of the cell. You also can see that we have a secretory vesicle indicated by the white circle, and inside that secretory vesicle we have several purple substances. The purple substances are going to go ahead and be considered secretory products, and these purple substances are going to go ahead and be released from the surface of the cell when the secretory vesicle binds and connects with or fuses with the plasma membrane. Again, this is exocytosis, where we're going to go ahead and have stuff inside the cell leaving the cell and being released to the outside of the cell. We also have something called endocytosis, and this is where we're going to go ahead and have stuff coming inside of the cell. Endocytosis is where we have engulfing of substances outside of the cell into a vesicle that is brought inside of the cell. And there are going to be two main types of endocytosis. What we're going to have, number one, is going to be phagocytosis and also pinocytosis. And phagocytosis is going to be endocytosis of particulate or solid matter, while pinocytosis is going to be endocytosis of liquid matter. So phagocytosis is where solid stuff is coming into the cell, and pinocytosis is where liquid stuff is coming into the cell. If you look at the diagram to the right, at the top you're going to see an example of phagocytosis where you're going to have particulate matter coming into the cell. Ultimately, you're going to have an invagination of the plasma membrane that eventually will go ahead and pinch off. And then you're going to go ahead and have a vesicle surrounding that particulate matter inside of the cell. And that vesicle will then go ahead and travel wherever it needs to go inside the cell to be used for its particular function. The same thing occurs with pinocytosis. Liquid particles or liquid molecules are going to go ahead and enter via an invagination of the plasma membrane that will eventually pinch off and form a vesicle. That vesicle then can travel inside of the cell wherever it needs to go so that it can be used for whatever it needs to be used for. Now, as I mentioned, membranes are going to go ahead and be semi-permeable. Membranes are actually going to be selectively permeable, or they're going to have selective permeability. And what we're going to see is a situation whereby cells will control specifically what is capable of entering and leaving individual cells. This is going to involve using proteins in the membrane for transporting substances across them. Remember, early on in this lecture we talked about the fact that in the plasma membrane we have several proteins, and that several of these proteins will be involved in allowing things to specifically cross through them. Now, we have something also called facilitated diffusion. This is where we're going to have movement across a membrane down the concentration gradient. And this is specifically where no energy is necessary to move things. They're going to simply move down their concentration gradient from high to low concentration. Facilitated diffusion is where you're actually going to have things not just passing through the membrane itself like water could, but you're going to actually have things passing specifically through a protein in a membrane. That's why we call it facilitated diffusion, because we're not simply passing through the membrane, but we're actually having to pass through a protein in the membrane. Another type of movement is going to be active transport. And active transport happens very frequently, but it's very different from simple diffusion or facilitated diffusion. Active transport is where we have movement across a membrane against the concentration gradient. In this particular case, a substance is going to go ahead and move from low concentration to high concentration. And to do this, we have to use energy. Energy is used to pump substances across these membranes. And in this particular case, active transport is used to maintain either high or low concentrations of substances, either inside or outside of the cells. And active transport can be very important for cellular processes that are going to allow our bodies to function. We're going to talk more about active transport in upcoming lectures.